Welcome, Andrew, to the Collaborative Research Center Origin and Function of Metaorganisms.
Thank you.
It's fantastic that you're here. You had great stories last year published, which got a lot of attention on the impact of microbes on human evolution. You want to tell us a little bit more? How do microbes affect our evolution?
Yeah, so I think the answer to that question is still largely a mystery. But maybe what we've discovered so far is that we've had a huge effect on microbial evolution within our bodies that could then in turn feed back and affect our own evolution potentially. So we've been able to compare the microbiomes in the guts of humans with the microbiomes in the guts of our closest relatives, the chimpanzees and bonobos, and then a little further out the gorillas, so the other African apes. And just the main result that we found is that humans have inherited many lineages of gut bacteria from our ancestors with chimpanzees and our ancestors with gorillas. And these bacteria have been passed down from generation to generation over millions of years and speciated alongside the human lineage. When we split off from chimpanzees, our gut bacteria split off from chimpanzee gut bacteria. So in a sense, our gut bacteria are part of what make us uniquely human. And the question is, are the bacteria actually driving evolution in humans? So are they acting as a selective force in human evolution? And that, to me, is very exciting. Still something we're trying to wrap our heads around to figure out how to study that exactly. But one sort of alarming pattern we found, too, is that humans seem to have, we maintained some of these lineages, but we've lost a lot of lineages as well. And something that's coming out now is that that loss of bacteria might be predisposing us to disease, like autoimmune diseases and metabolic diseases.
There is a human-specific core microbiome which distinguishes the Homo sapiens from the chimpanzee and the orangutan and the gorilla, etc., etc.
Right, exactly.
So it's a little controversial in the community because how stable is a microbiome? And is there something like a Homo sapiens core microbiome, given all the dynamics, which is food dependent, environment dependent, individual specific, etc., etc.? Would you explain how you define this core microbiome and what are the evidences that humans, despite their dynamic and changeable microbes, have microbes which allow you to say this microbiome comes from humans and not from chimpanzees?
Right. So it's, I think you're totally correct that the human microbiome is very dynamic and can change with diet or any sort of environmental difference among humans can affect the microbiome and create a lot of variation in the composition of the microbiome. But what we found is that despite all of that variation, there are some bacteria that are always with us, with humans. So we find them in all human samples that we look at. They might differ in their abundances, but they're always at least present in humans. And when we sequence the DNA from those bacteria and compare it to the DNA sequence from the bacteria of chimpanzees and the DNA sequence from bacteria of gorillas, we can build phylogenetic trees of the bacteria. So actually reconstruct the evolutionary tree and the evolutionary relationships among the bacteria. So when we do that, we find that the evolutionary trees of the bacteria line up exactly with the evolutionary trees of hominid species. And so from that congruence between the phylogenetic trees of the bacteria and the hosts, we can infer that there's been this long, stable association over evolutionary time. So I think that's the main line of evidence, is this phylogenetic evidence.
Mm-hmm. The obvious question, of course, then is, do these bugs make us human, or what is there a human-specific influence? What is human, and which role do microbes play in us being humans? That's the $100 million question, of course. And I can come up with many features of humans, behavior, etc., art, cognitive capabilities, which distinguish us from great apes. Mm-hmm. Do you really see an impact of microbes on that human-specific behavior or humanness?
It's possible. So we know now, through these experiments with germ-free organisms, like germ-free mice, for instance, that the bacteria are playing a role in essentially every phenotype that's been examined. It might be a small role, it might be a very big role. So if we can learn anything from mice about humans, it implies the same might also be going on in humans. But the problem with studying in humans, it's very hard to do manipulative experiments. In an ideal world, you could have a germ-free chimpanzee and give it human gut bacteria and ask, does the chimpanzee become more human-like? We can't do that experiment with apes, but we can do that same experiment with mice. So that's exactly what we're doing now. We're taking different mouse species who have similar patterns in their gut bacteria. The gut bacteria seem to evolve with the mouse species over time. And we can, in germ-free mice, swap out the gut bacteria of the different species and look at the effects on the hosts. So hopefully with this approach, we're going to be able to actually show that gut bacteria are playing a role in generating phenotypic differences among the mouse species. So that's sort of the future I think I'm the most excited about.
Yeah, but this is already, I mean, the Bienenstock group and others have clearly shown that germ-free mice, for example, are more anxious
Yes.
than wild-type mice.
Right.
And so I want to come back to the impact on the humans. So would that imply, or would you want to say that taking away microbes makes us, or the loss of the diversity which we face in humans, according to your work, makes us changing this type of behavior, for example?
It's difficult to say conclusively in humans because of the lack of experimental manipulation. But one thing that can be done is sort of looking retrospectively at, say, people who have taken antibiotics early in life, people born with cesarean section, and looking at later in life, do they have different behavioral phenotypes, different health outcomes, and so on. So I think that sort of work is being done now, and there's evidence that you're more likely to have asthma, for instance, if you had a C-section or had antibiotics early in life. So the question is, does the variation within humans and levels of diversity, for instance, correlate in some way with behavioral phenotypes or more complex phenotypes? And I don't think we have any strong evidence that that's true yet, but I don't think people have looked either. So it's future,
you
something
consider
to do.
that as possible?
I think so, yeah.
Okay,
yeah.
I mean, another major outcome from your studies was the surprising finding that obviously humans have lost a number of microbial lineages and key species which are present in the great apes. I mean, on the other side, we really have to move from correlation to causality. And so what do you think, what does it mean for us being humans that we all of a sudden now have a much lower diversity in the microbiome than our closest ancestors?
Yeah, so I think of the loss of diversity from humans, it's a little more complex than to say that all humans just lost a lot of bacteria. So if we look across different human populations, we see different degrees of loss of bacteria. So if you look at all human populations, the most rural human populations living the most traditional lifestyles, hunter-gatherers, for instance, versus humans living in urban centers and cities, all humans have less diversity than African apes. So that implies that there's been some loss of diversity in humanity's deep evolutionary past before human populations diversified. So that loss predates differences among human populations. But if you look within humans in the United States, for instance, in urban centers or urban centers in Europe, westernized populations, we see a secondary loss of diversity. So they have even less diversity than the ancestral human state. And so that first round of loss of diversity that happened early on in human evolution seems to reflect the transition of humans away from eating a lot of raw plant material towards cooking food and eating meat. So a lot of the bacteria that have been lost, we know they're associated with the digestion of plant polysaccharides, and some of the bacteria that have increased in abundance we know are associated with meat eating. So it's possible that that change in the microbiome had some role with our dietary evolution, which we know, or we think, played a role in the evolution of human cognition, perhaps. So it's possible all these things are connected. The second round of loss that happened in urban centers or in westernized populations is largely thought to be deleterious or have a sort of pathogenic effect. A lot of modern diseases seem to be able to trace their roots to imbalance in the microbiota, so autoimmune diseases and obesity and so on, these things that have been called modern diseases. And that idea is also termed the hygiene hypothesis, that we're sort of overly hygienic, too much antibiotics, not enough dietary fiber, not enough bacteria, and it's hurting our health.
So many anthropologists and our behavior colleagues tell us that human and also grade A behavior, which is very social behavior, has all the features which allows to transfer microbiomes between different members. And so you came up with a surprising statement that there is a certain type of heritability in the microbiome. Can you bring that together with the social behavior? I mean, could that be the mean how microbes are transferred?
Yes, so we have some evidence of that in chimpanzees. So we did this study in Gombe, Tanzania, which is where the population of chimpanzees studied by Jane Goodall lives. So it's a very well-studied chimpanzee population dating back decades. Primatologists go out and they record the behaviors of the chimps every single day, who they're with, who they're grooming, who they're around in proximity to. And we were able to show that chimpanzees that come into social contact, and the more social contact the chimpanzees have, the more microbes they share with one another. And also, the more social chimps were, the more diverse their microbiomes were. So the more species they harbored. So it's possible that humans, that might be one of the causes why humans in urban societies have lost so much diversity. We are a little more isolated from one another. If you compare social interactions in cities versus social interactions in, say, hunter-gatherers, it's like night and day. So
the transition from the ancestral beings to the humans would be a kind of a reduction in the social behavior, and that's what you could imagine that happened?
Yeah, it's possible that this change in social behavior might be contributing to it, at least in part.
Could one turn the argument around and say that selection drive for evolving this type of social behavior was to spread the microbes among a given population? Yeah,
so if you accept that having a diverse microbiome is beneficial and harboring these microbes helps your fitness, and you also accept that social interactions help you to maintain this diverse microbiome, then logically the very next step is to say that, well, perhaps selection has favored social behavior because of the microbial benefits that it confers.
Would there be an experimental approach to test that? I mean, in mice or autistic mice versus social mice?
Yeah, so I think it might be difficult to test experimentally in an animal due to the timescales it might require for selection to operate and for you to see an effect. You know, for a PhD student setting out to design a thesis, I would not recommend such an experiment that might take 20 years. But it would be possible, I think, to look at this sort of retrospectively in an evolutionary context. So, for example, comparing social and asocial species and comparing the microbiomes of the two and seeing if the social interactions are correlated with any sort of microbial features that also increase host fitness, would suggest that perhaps selection for microbial transfer was underlying the evolution of sociality. Of course, social behaviors confer all sorts of benefits beyond just transferring microbes. So it's also likely, I think, that selection is favoring the evolution of sociality for many different reasons all at once. It has many benefits that are non-mutually exclusive that can all be selected together.
So, summarizing this part of our conversation, my many anthropologists and paleontologists are now very keen, due to modern technologies, ancient DNA and new findings of human remains in different places, to reconstruct the human history. Would you and your research and your microbiome-oriented research give them the message there is more than just the human bone, and maybe the associated microbiome must be considered when we really want to understand human evolution?
Yes, I think that I maybe would say that. I think understanding human history and human migration, the history of that, can probably be done with just the human genome by itself. But if we want to understand how humans are adapting to pathogens or to the beneficial microbes that live within us, we also need to know what those microbial populations look like in the ancestors. So studying things like coprolites, these partially semi-fossil fecal materials, and also dental calculus and things like this, it's going to be a huge field, I think, in the next 10 years. There's already some work that's been done in this to understand, for example, the evolution of the plague, Yersinia pestis, using dental samples from thousands of years ago. But as far as I know, studying the full populations of commensal organisms like the gut microbiome is still in its infancy, I think, from ancient DNA studies. Yeah.
Fascinating. You briefly recommended that a graduate student should not hook up on certain projects which take about 20 years. Right. Of course, we fully agree. But that brings me...
It's too bad in a way. Yeah.
That brings me to quickly going over your own career, which is an astonishing junior researcher career. You started a little more than 7 years ago. You started your PhD in Yale, finished that with a tremendous set of papers, which high impact and which now redirects a large part of the community looking into things which we haven't seen before. Meanwhile, you are a distinguished postdoc at Berkeley, and you have already the offer, and you have accepted the offer to go to one of the Ivy League universities as a professor to Cornell University. I mean, which factors have contributed, or what do you think was supporting your really quick academic career?
Yeah, I think I've been very lucky, not to totally write it off on luck, but I really have been very lucky. So I started out in college, I was a music major. And for the first 3 years of college, I played viola. I wanted to do viola performance. That was my idea.
That's fantastic.
Yeah.
I didn't know.
And I read The Selfish Gene by Richard Dawkins.
Yeah.
And I grew up in Tennessee and hadn't really learned about evolution. Tennessee's in the South. It's sort of more conservative. We don't really learn about evolution. But when I read The Selfish Gene, it was like a light switch flipped. And from that moment, I knew I had to do this career. And so when I was a junior in college, I switched majors to biology, and I've been all in ever since that moment. So I think that's an important thing. You have to be all in a sense. But beyond that, there's a lot that's out of your control in a sense, or that relies on other people, which I shouldn't say is out of your control. You have some control over it, but it relies on other people. So at Yale, I was advised by Howard Auckman, who you mentioned previously, who was a perfect advisor for me. He's very critical person, but also very supportive, and we worked very well together. So that has been maybe the most important factor in my success so far. What I've had so far is finding a good PhD advisor. So if anyone's interested in starting grad school, I would say that's the most important thing, is find an advisor who you can work well with, and the most important thing is that they're interested in what you're interested in. So you have to have a shared interest, and so that they can support you. The other thing I would say is just to always be opportunistic and never be overly negative. A lot of people in grad school can get themselves into a hole of negativity where they are convinced things aren't going to work, or that they're going to get scooped, or that the list can go on and on and on. You have to be ambitiously optimistic, I think, all the time, and also opportunistic. So if you see an opportunity for an interesting project, or for sort of piggybacking a project off an existing project, just go for it. try to have a lot of things going on at once, and one of them eventually will work out. I think it's a lot of luck. You have to kind of stumble around to find something interesting, but you can control how fast you stumble, if that makes. That's sort of how I think of it.
Yeah.
So it's good to stumble quickly.
Yeah.
And eventually you'll stumble into something.
Yeah.
Yeah.
I think this is. I think we all can agree, being open-minded, and but then having the right persons around you. But you also have to have the antenna for these persons. It must be a dialogue. So, coming back to your comment on that you started actually as a music student. Many scientists are musicians, and in their previous life, and they still love music and get inspired or driven by music also in their scientific thinking. Do you see a place for music in your life, in your scientific life?
Yeah, so music, I think, for me, it's like a meditative way to relax and to shut off this sort of verbal chain of thought that your consciousness is. So it's very easy if you're stuck on a problem or stuck writing or something, you can't get out. But if you can sort of reset, music is a great way to reset in that way. So it sort of relaxes and turns off the thinking part of your brain for a while, and you can think in a different way and then maybe come back. It's refreshing. But yeah.
Where next? I mean, you opened a new window. You are supported, of course, by new technological breakthroughs, which allow now to look very deep back in time, but also very deep into tissue and cells. And so where do you want to go next? And if you look a little bit more broader, where you think the host microbe community should go or must go or will go in the next 5 and 10 years?
Yeah, that's a great question. So for me, I think the most interesting next set of questions is to try to understand how has the microbiome shaped the evolution of animals. So we now know that animals have very clearly shaped microbial evolution, but the other side of the coin, how have microbes shaped animal evolution, is still, I think, a big mystery. So using these germ-free animal models, like germ-free mice, we can start to ask how different microbial assemblages that have been produced by evolution affect phenotypes in the hosts, and to hopefully understand how all the great diversity of animal forms that exist, what role have microbes played in producing those forms. So that's sort of the next stuff that I'm personally interested in. But I think the field at large is, there are many directions the field's going to go, but I think that's also one of them, trying to understand how microbes have driven the evolution of humans and other animals.
There's major interest now also from industry. Industry gets very much aware about that there may be a neglected part of our health, which essentially can also be manipulated. Do you see there, is that an interesting area which will develop and where young people may end up?
Yeah, I think so. So we were all told when the Human Genome Project was happening that we were going to cure everything.
Nothing happened.
And yeah, maybe a couple of things, but basically nothing, right? And the reason for that is because you can't change the genome. You can learn, oh, you have that mutation, and so you'll have this genetic disease, but there's nothing you can do about it. But with the microbiome, well, maybe now with CRISPR you could do something with it, but with the microbiome you can actually manipulate, like you said, you can change the microbiome. So I think we're already seeing this some with like Clostridium difficile infection being cured by fecal transplants. We're only going to see more and more treatments that are targeting the microbiome. So, for example, one, I think, pretty fascinating subfield that's been created is this idea of psychobiotics. A probiotic that can change your mood and maybe prevent depression or prevent anxiety. We know the microbiome plays a role in in moods and behaviors. So if you can manipulate the microbiome to improve mood and behavior in a way you'd like for yourself, why not, right? It's very exciting. But I think the list is virtually endless for the types of diseases that might be treated or potentially even cured by the microbiome. Autism is another great example where a couple of papers last year showed that if you fix the microbiome in an autistic mouse model, you can alleviate the symptoms of autism, which if you had told people 5 years ago that that were true, they would have laughed at you. And so who knows what we'll find.
Yeah.
Yeah.
A last aspect which I find interesting in your career and in your research is there's not so many people which really combine hardcore fieldwork and hardcore lab work, and because the culture, the thinking, how you design the experiment is very different if you go in the jungle and collect the feces from the chimpanzees and.
Right.
And you mentioned the gorillas. You mentioned the gorilla station from Jane Goodall. I learned recently that this station, actually, the gorillas there are kind of already cultivated. They are not really wild, wild anymore. So, is that a particular challenge for you, doing at the same time field work and lab work, or is that a logic combination? You want to test some observations from the field by controlled experiments in the lab, and how you see that?
So I think now and in the future, I'm really focusing on experiments more and more all the time. And I can see that continuing for quite a while. But it's essential, I think, to look out in nature and see what actually is in a sort of exploratory way. I think there's a real value to natural history in that sense. So the history of biology is interesting. Since the 60s, I think there's been a very strong emphasis on strong inference. So there's that famous paper by Platt back in the 60s that argued that the only way to do biology is to design alternative hypotheses and then exclude one and then move forward down this logic tree until you discover something. But nothing can replace going out into nature and looking with a new lens. If there's a new technology that's never been applied to the study of natural history, go out and apply it, and you're bound to find something exciting and new in a very exploratory way. So I'm a big fan of exploratory science. I think it's not discounting the importance of that sort of strong inference approach, but I think there is a tendency of people who focus on experimental biology to sort of dismiss natural history, and I think that's a mistake too. So I think both can go well together, yeah.
Yeah, for sure.
Yeah.
And isn't it interesting that there's a paper came out recently which calls all these modern diseases nature deficit disorders?
I haven't heard that.
Yeah, so I think I also find that maybe they mean something different, but that describes very well the trouble of our time. And also maybe in research, not too many people really go out again in nature and let them get inspired by what happens in real nature. And,
There's a course at Berkeley, a field course called Ecology, that takes students from California out to the field and out in the desert, and they camp and they see the stars and they see the rodents running around. And many of them are from LA. They've never been outside the city in their whole life. And you can see the look in their eyes. They didn't realize how amazing nature can be. And so I think everyone should have that experience. Yeah, it can be life-changing.
Thank you very much, Andrew.
Yeah.
This was a pleasure to have you here,
Thank you,
we are looking forward for some more time with you. Thank you so much.
Thank you.
Thank you.